Welcome to the Biotech Startups podcast by Exceeder. Join us as we speak with first-time founders, serial entrepreneurs, and experienced investors about the challenges and triumphs of running a biotech startup, from pre-seed to IPO, with your host, John Chi. In our last episode, we spoke with Terry Lowe about his transition from big pharma to diagnostics, the challenges of moving into the diagnostics industry, and the contrasts between drug and diagnostic development processes. If you missed it, be sure to go back and to give part 2 a listen. In part 3, we talk about Terry's introduction to Akoya Biosciences, its strategies post-merger, and subsequent focus on spatial genomics, the challenges of scaling up a business, and Vizgen's platform, mission, and future developments.
So I think, and not to speak for Akoya today, but certainly at that time, we were trying to revolutionize this field of spatial biology. Like, nobody really knew about it. It's kind of still kind of not a familiar term in the field. And this concept of spatial biology, which is again really kind of looking at what's in the tissue, using molecular markers to identify those cells that are in the tissue, and then figuring out how they're interacting to better understand the biology. The strategy, again, was one, is kind of continue to push this into the clinical side with that PerkinElmer platform, right? So it's very embedded in what we call the translational space, which is kind of like in between research and clinical, but doing clinical research, and then trying to take that into the clinical field. But also because there's a platform that can do much higher protein detection, that was more of a research type of platform. So the idea at the time was that you can cover sort of the spectrum of sort of earlier research to translation to all the way to the clinic once you get diagnostics approved out of it.
Excellent. And that, that sounds like a lot, like for a company that is 50 people strong. So that sounds like a lot of initiatives, and there's like the kind of exactly what you said, like translation through the clinic, and you're covering the spectrum. At the time when were all of those just like the key initiatives, like for as president?
There's also the whole company building process, right?
Yeah.
It's okay. Like, how do we establish international, subsidiary?
Yeah.
How do we get our supply chain going, and how do we manufacture product? So 50 people, most of those are, some R&D and some salespeople and support people, right? But
Yep.
The other infrastructure part didn't come with us, right? Because those were not easily separated out. So for a year, we had this sort of transition services agreement with PerkinElmer, where effectively we're paying them, for services to help support our business. So now we're separate companies, but they've been supporting this business all along for however many years, so shouldn't be too difficult. But now that we're two different companies, it's a little bit arm's length, and so we had 1 year in order to kind of extricate ourselves out of there, and then build out our own infrastructure to be able to do that. So it's so that was a big piece of it, is just scaling up the business. Today the company's, I don't know, like 300 some people, 350 people, but it's certainly grown tremendously, I think, from the early days.
Absolutely. And yeah, that is an absolutely big component, is like not only the science and the products, but just like company building. Like just like it is.
Yeah.
It is no easy feat. It is a lift. That is for sure.
Yeah. There's so many different functional areas within a company, and you have to cover them all, right? There's you can't just leave one out, and then you need it all. Like,
yeah. We're just like, well, we'll just like address that later. Like, finance, maybe we'll just like address that later.
Right.
So after Akoya, when did you know it was time? It was time to leave Akoya and join Vizgen.
Sure. So in the spatial field, so saying most of the focus has been on protein, and that's sort of been the legacy of and at the time, this been going on for a number of years. I always thought like spatial meant protein. Like it didn't even occur to me that this could be done in genomics, or in this case, RNA. And, when I started to see some of the papers coming out of Harvard University on something called MERFISH, that was such an eye opener. And I think there started to be a real buzz in the field about like, wow, this is something that can really address the genomics field, but having spatial with a high number of targets and high resolution. And that's just something that had never been done in the genomics field, or in the spatial field. So this new area that we call spatial genomics, which is a subcomponent of spatial biology or spatial omics, that was the opportunity that I said, like, this is going to be massive. Like, if this could actually work, like for the first time, I actually thought something could work, that would be just amazing. So that's why, I had started talking with Vizgen about coming over. And by the time I came over as CEO, the company had been about 1 year into it. Like they'd been founded the year before. So I would think I was maybe somewhere between 12th and 14th employee of the company. But when I was interviewing earlier in the year, I was like, there was only 5 employees at the time. So that's how much earlier, like now it's like really, really early, we're talking about from a company standpoint.
Yeah, that I mean, yeah, that like, 5 people, like that is a very drastic transition and much more company building kind of taking place, and probably at a very rapid clip. Can you talk a little bit about that, like that early team, the little bit of Vizgen's history and kind of the team behind the technology, as you joined with a 5-person team?
Yeah. Yeah. So as you can imagine, I mean, the core of this really is R&D, right? It's like brought this technology over from Harvard. Now we need to develop this, turn it into a product. So we call that technology MERFISH. And then very rapidly, we developed a product which we call MERScope to be able to commercially launch it. And the heart of it was really just having this core R&D team to be able to rapidly turn this technology into something that we could sell to customers. So I joined, I think, in September of 2020, and then, by the beginning of 2021, we had like 17 people. I think the next beginning of 2022, we had like 60 people, and then the beginning of this year, we had like 150 people. So it became a very drastic scale up, particularly for a tools company. I mean, it's not, some other industries, I think you can build really fast. But this was a pretty rapid build out. And we actually took that technology out of Harvard and then launched it, first as commercial pilot within less than 2 years. And that is, incredibly fast from a commercialization standpoint. And so by the 3rd year of the company, we had, our first full launch year of the product. And I think we had sold, we had an install base after the first year of like 70 instruments. So it was a pretty great, kind of journey in terms of like scaling and building up incredibly fast.
And, with a journey that fast, it kind of feels like building the plane as you're trying to, at least from Exceeder's, our experience is kind of like that experience is like building a plane as you're trying to fly it. And, maybe just to kind of set the table for folks who are maybe not as familiar with the spatial omics kind of field. Could we like zoom out for a moment and just like learn from you, what is the current state of like spatial genomics, like as a market, and why is it that way?
Yeah. So I think when Vizgen launched MERScope, kind of at the end of 2021, beginning of 2022, I would say that was really the first spatial genomics commercial platform that we call high-plex, meaning it can detect a lot of RNA, a lot of targets, and also high resolution, which means you can get it resolution at single cell or subcellular level to be able to detect. So, that's why there was such a huge, I guess, excitement and demand at the time, because this has never been done before. And this is like brand new, like researchers out there for the first time are being able to generate data that they'd never seen before. So that was in 2022. There have been a couple other platforms that have come out at the end of 2022, so about a year later. And I would say the market today is really just these three platforms right now that are out there. And so we're still in the very early innings, right? It's like this is like literally has been, less than 2 years since we launched. So, researchers are still kind of starting to build out, I think data and generate data from these types of platforms.
Absolutely. And you're with a market that is like very early innings like this, and there's only a handful of platforms. Can you talk a little bit about Vizgen's platform, your guys' mission and focus, and kind of what is Vizgen's like approach and strategy in terms of like this go to market in this kind of more nascent budding industry?
Yeah. I think, we have a certain advantage in that, our kind of core technology, which came out of Harvard, that really was the established kind of approach in terms of setting the blueprint, like how do you do this type of thing? And I think others have kind of learned from that, and they're try to develop their processes and products kind of based on some of that blueprint. Um, what really I think distinguishes what we do and what we want to do as we move forward is that the data quality itself is kind of the primary goal here, right? Which is a lot of what we're learning right now from being able to use a platform like this is really fundamental biology. Like we're learning about like what cells are located in the brain, what cells are located in different types of tissue or in cancer, or as we develop, as organisms develop, how are those differentiated? So understanding the detail of that at that molecular level, but also making sure that we understand it and get it right, right? Because
Yeah, yeah.
there's a lot of potential to kind of get noise, or it's hard to know sometimes what is ground truth, because this is a new technology. There's not a comparator, right? It's not like this is something that we can compare against something else that's been very well established. There are other kind of orthogonal platforms that you can kind of compare to, but fundamentally it's like this is new stuff. So making sure that when researchers are being able to use and generate this kind of data, that they're kind of getting the data right. That's the target, right? That's the goal, and not as trivial as maybe as it sounds, right? It's like there's a lot that goes into it. So in a 1 centimeter square area, I mean, you're trying to detect more than 1 billion RNA molecules and discriminate them within 50 nanometers of each other. So it's a lot of precision, a lot of detail that goes into that. So it's not easy to do, and so that really is the focus, is really to make sure that we're generating the best quality data that's out there, and then also making it, I think, as accessible in terms of being able to use this, for different types of research. So not just one specific application or one specific area or one specific organism, but it's just like sequencing in that sense, right? You want to be able to use it everywhere that you can imagine you've got gene expression happening. So that's I would say that's really the direction that we're trying to drive the platform.
Totally. And yeah, I can imagine for anyone who's like thinking about adopting a platform, it's like, of course, it's critical. The data's got to be good. You said it so eloquently. If I'm a lab considering a platform, it's like, yeah, like I need to make sure that this readout I can rely upon it.
Right.
Which it sounds like a hard, especially for new technology, that seems like it seems, because right now, if you're thinking about it from other instrument platforms, you're like, yeah, real-time PCR. Like, we've been doing it for a long time. It's very,
Yeah.
very easy to communicate. In this instance, it's like exactly what you said. There's like not so much of like a control group which you can compare against. You're just like, here's here it is. Like, and so how does that, how do those like the comms like unfold for you, and how do you approach that?
Yeah, so it is challenging because it's not as an easy thing to communicate or even demonstrate in some cases. And it becomes very, very technical. And there's a lot of bioinformatics analysis to say, hey, how do we show that something is more accurate, or that it has higher sensitivity, or this is real signal, and it's not,
Yeah.
it's not noise, right? That that is not an easy thing to necessarily explain, and also to conduct the analysis is fairly complex and sophisticated. So, it's much easier sometimes to say, hey, I can, instead of 1,000 genes, I can detect 5,000 genes, right? That everyone gets that, right?
Yeah.
Okay, so,
Yeah.
like, that must be good. Like, we're
Yeah.
doing more, so that's better than 5,000 is bigger than 1,000. So it's a much more subtle kind of way to message, and also to be able to educate on. So it's a process, and I think it just takes time, and I think it's also, as researchers getting more experience with it. Again, it's like the first time that they're really kind of being able to see and access and look at this kind of data, and so it does come out over time. But of course, we would like to get that sooner rather than later. So yeah, I mean, I think that's kind of the state that's in. There's also, I think, that there is some level of, okay, you know, in some cases, like, this is good enough. Like, I don't need the best data. I just need data that's like good enough for what I'm doing.
Yep.
But I think the challenge of that right now is that until we really have like gold standard and very clear, like, what is truth, what is ground truth, then you can kind of move off of that, right? Because then you can kind of say, hey, it doesn't really matter. I'm a little bit off here or a little bit off there, less sensitive or less specific. If you're until you really kind of really get the I would say the kind of the core foundational science down, like, you want that to be right and as accurate as possible. And then from there, you can kind of say, all right, for this particular application, it's not so important that I know, this level of sensitivity for my detection, because that's really not relevant for my application. But you don't really know that until you kind of know sort of the foundational biology piece first.
Totally. Yeah, it's kind of like getting that just a large enough sample size where there's like, okay, this is baseline. Like, there's like
Exactly. You need a baseline, and that's, in some sense, that's a lot what's going on in spatial today, which is, probably people are familiar with things called cell atlasing, and that is really trying to create these maps. And you could kind of think about it like, if Christopher Columbus, if he actually had a map of where he was going,
yeah,
he probably wouldn't end up in the Americas. Like, he would have
yeah,
known that was like not the right route if he wanted to get to China, or if he wanted to get to India. Like, he didn't have a map.
yeah,
So the same analogy is, look, like, if you want to know what are the right targets, or you want to have a map so that you can figure out, like, what is happening there that's driving disease, what is happening there that I can target, that potentially will allow me to stop the progression of that disease. So that level of biology, I think it all starts with having the right sort of mapping. And that's actually, I think our vision statement is that, every disease has a map, every cure has a path. And one of the things that are very active right now is these cell atlasing programs. And one of the best examples is that there's a lot of work being done on brain atlasing, right? Because the brain is just a very highly organized organ, and you have very specific types of cells that are located in different parts of the brain that serve different functions. And so it actually makes a great map. Like, you can
mm,
actually identify, like, there are these different cells, and you figure out what those cells are so that you can understand what their function can be. Um, and the next level beyond that, though, is really to kind of understand how different cells are interacting with each other, right? So now I know this cell type is next to another cell type. What kind of influence does that have when they're next to each other, and how do they interact, and how does that change the expression maybe of other genes, which then relays downstream activities? So this is kind of the beginning point of kind of really just understanding, again, some of the basics of biology, right? And where that helps lead us to is then looking at disease, right? Things like, like, we don't understand how Alzheimer's works, right? So why is it we can't develop drugs that are effective against Alzheimer's? We don't really understand the disease mechanisms there. So this is why there's such an amount of resources now being put into this kind of cell atlasing to kind of understand the biology first.
Very cool. It just kind of goes back to the kind of point we were talking about earlier about just having this increased understanding instead of just like forcing mechanism of just like, let's just do more volume and hope we find a hit. Like this is like, okay, let's actually figure out
Exactly.
and try to be more knowledgeable before we go on this road trip. Like let's try to, and then from there we can probably be more, more efficient in our approach.
Yeah, that's exactly right. I mean, we don't really understand, what's happening, and so we're just taking random shots, right? And
Yeah.
we can take enough random shots, and as we go through that process, we'll find ones that work without really being able to really map it out and say, look, this is what's happening, this is what we should be targeting, this is why this works. We can demonstrate it, and we can visualize it. So I think that's the difference, and hopefully this type of technology can be adopted fast enough so that maybe in our lifetime we can actually see the downstream benefits of what this data and what the science generates.
Very cool. Very cool. And to kind of, as we kind of talked about, like there's the aspect of the science of, a company, but there's also the company building aspect of
Yeah.
running a business. And, the past couple of years, it's, we've lived through what feels like a lifetime. 2020 through 2021 was this whole thing. 22 to 23 was this whole thing. So how did you navigate these kind of like different periods for Vizgen, and what's your philosophy around kind of like capitalizing a business and running a business through these kind of like cycles?
Yeah. So look, I mean, I would say this is a never-ending learning process. As
Yeah.
we go through, every situation is unique. I think this cycle has been incredibly challenging and more challenging than probably most of us have seen in recent experience. When we were in sort of that 2020, 2021, which is ironically, at this kind of height of COVID, but at the same time, this
Yeah.
is the financial markets were kind of taking off, and all the company valuations were through the roof. And so, the concept in those days was really about, hey, money is free, basically, because you can just access money so easily, and you really just want to drive as much growth as fast as possible, right? And then, as we got into 2022 and now 2023, it's completely flipped, right? Where it's really now the whole focus broadly across the financial markets is that, look, we want companies that can generate profit. So don't burn a lot of cash, don't spend a lot of money. It's more important that you can be efficient with what you have rather than, just kind of growing at all costs. So, I think that's a transition that almost all companies have had to now experience to go through. And I would say that 2021 period was just it was kind of a great time to be
Yeah.
in it. But unfortunately, it may not happen that frequently. So yeah, so I think we just have to all kind of work on sort of being able to be very adaptable and just recognize, look, this is the new reality we're in, and we're going to have to, work things and have strategies that are a little bit different than before. Like if we thought we were going to spend all that money, a couple of years
Yeah.
ago, like we're not going to spend that money, in this market and in this environment. So, yeah, things definitely become a much tighter and much more focused around efficiency than it is, let's say, just around, hey, let's just drive growth, and
Totally.
that's the metric, right?
Yeah. Absolutely. And obviously, it's still very early innings for Vizgen. And we're entering 2024 soon. Kind of doing a little bit of a retrospective and then a prospective. On a retrospective, just like thus far, what were some like big challenges and triumphs for you,
Yeah.
at the helm of Vizgen?
Yeah, I mean, certainly the financial markets themselves kind of create a different dynamic in terms of, when you're a private company and you're not a profitable company, you're always in some mode of fundraising, right? Like, because you don't make money yet, right? So you need money, and you end up having a budget that will last you for a certain amount of period of time, and then you got to raise money again. And it's kind of like, you're still, living with your parents, and you're kind of relying on an allowance, right? to
Yeah.
get to get
Yeah.
through.
Yeah. Yeah.
You
Yeah.
don't have your own job. You're not like self-sufficient yet.
Yeah.
So I think that's one of the things that, even though we're a very young company, and it's kind of, this is what you would expect with a company this young, just finished our fourth year, right? You're not going to be profitable. You're not at a point in maturity of the company where you're actually, generating positive cash within the company. So, just kind of trying to navigate and manage through that, I think certainly is a challenge. I mean, certainly for Vizgen, but also, like I said, most companies that are kind of in the private and, startup world, are going through that. We also grew, incredibly, as I said, rapidly, almost unprecedented. If you look at life science tools, like how fast did this company scale up? If you look at our revenue trajectory, for this year and last year, I mean, it is, really, you don't find other life science tool companies that have generated this much revenue in such a short period of time, right? Just this type of market adoption so fast. We've been working on, continuing to build out, scaling up the business, the organization. Like this facility actually right behind me is not our headquarters. This is actually in Waltham. So our headquarters in Cambridge. But we got this facility built out and moved into this summer, just for the operations side. I mean, that's, from going to no operations to having its own building. That's kind of how we've kind of been transitioning so quickly. So before that, like, we were literally packed into one laboratory between R&D and manufacturing and QC. And now we actually have, some elbow room for people to kind
Yeah.
of build things and do things and
Cool.
put instruments in there. So so the growth has been a lot to manage, and so we're certainly, as any young company or as any young person, you're going to go through those growing pains. So it's no shortage of things to work on and improve upon, and I think that's what we've really have been doing in 2023.
Very
Yeah.
cool. And looking forward, like 1 or 2 years out, what's in store for you and Vizgen?
Yeah. So one is I'm super excited now that we're kind of, I feel like we've kind of turned the corner on some of the, infrastructure building and kind of getting us in the right place and trying to get also the product itself. Like there's always a maturation of the product, right? It came out very early. We continue to kind of mature and make sure that we can make it, consistently, that it can work consistently. And I think we've now at a stage where, we're really going to be pushing forward new developments in our product pipeline. And that's really where I think it's going to be exciting to see in 2024 as, these capabilities, when we first came out, like, wow, that was, incredible and exciting. And it's the first time people could do it. But, 2 years later, 3 years later, okay, like what's next? Right. So people are hungry to kind of continue to move on and move quickly, particularly in a field like life science research, because what researchers are all about is that they're trying to publish, right? And to publish, you need to have something new. You need to do something that other people haven't. And having the best tools, having new capabilities where people couldn't do something before, that gives them an advantage to be able to kind of create, to do the science, right? To kind of get those insights. So yeah, so we have a big focus on really kind of pushing out our product pipeline and new developments. I really can't talk too much about sort of what we're doing because we haven't publicly announced
Yeah.
that next year, but yeah, but we're pretty excited about that.
Sounds like there's some cool things in store. Even, well, until it's officially announced, we'll wait till then. But
Yeah,
that sounds very exciting.
yeah,
And
no, that's
and
really great. Yeah,
and I agree. Like I think, you know, you really, hit the nail on the head. It's just like, in order to publish, it's got to be novel. Like, and part of that is making sure you have the right tools. Exactly what you said. Like, if you don't have the right tools, it just becomes just like shooting in the dark, which is a much harder and we shot in the dark for a very long time, but now, at least now, it's like we're actually illuminating it and being a little bit more precise in our approach.
Right. I mean the short of it is now we can actually visualize things at a molecular level, right? So when we're creating, instead of just looking under the microscope and saying, "Hey, I can see the tissue," like you're literally like able to look down and look at, the genes, the RNA that's being expressed, the different proteins that are on all the individual cells. You have that level of molecular detail that you can now map out in your tissue, and then you can basically do all of the analysis you could ever dream of to kind of figure out like how does that all work together and how does that fit together. So it is a definitely a different type of stage I think that we're in, and from scientific discovery standpoint, and I think it's just incredibly exciting to be part of that right now.
Likewise, and almost whenever I like learn about this and hear about this, it feels like the science fiction future is now. because sometimes I'm just like, I do remember just being in the lab and just like, yeah, like that's not really high fidelity, but I get the general, I get the kind of like high level gist of it, but the precision and the granularity which is like not there, and to like think about we're living in a time where you can get that into that level of like precision at that level of like kind of granularity, it just like blows my mind.
Yeah.
And as kind of a traditional closing question, and Terry, thank you for your time. We always like to round it out here with just two questions. And the first question would be, would you like to give any shout outs to anyone who's supported you throughout your career?
Yeah, I think the one person I'd probably highlight the most is my dad. I mean, he's also the one who wanted me to be a doctor, but I think I think once he got over that, he's been really really supportive, and he's just he's always been such a wise person in terms of being able to provide advice and things to really think about. And when I talked about career planning, I mean he was very much instrumental as part of that. And I think you can see is like a lot of stuff kind of has played out in a lot of ways that I had planned for, and it was really kind of a testament to a lot of what he had kind of educated me on, and what his feedback was on how to think about things. So I would say a lot of credit certainly throughout the years to my dad.
Awesome. And likewise for me, I think the familial support is just so critical.
Yeah.
Especially in the early days where you're just like, what can feel like you're just like aimlessly, you're just like you're trying to figure it out. Having that guidance is so imperative.
Yeah.
And so I feel the same way.
Yeah.
And this, oh, go ahead.
By the way, and I can't leave out my mom, because if she listens to this, then I
yeah. Sorry, Mom.
So good job, Mom.
Yeah. Totally. And then the second question, if you can give any advice to your 21-year-old self, what would it be?
Yeah, I mean, I think I would just tell him, look, stay the course. Things are going to work out. It hasn't ended too badly. Keep on going and just be confident and what you're doing and what you're about. And I think that's, I would say the one thing is like, just be confident. I like it. It does work out, right? It does. You get to the place where you think you should be, and things kind of work themselves out. And so just be confident in terms of, how you're going about things.
Absolutely. And I think, thinking about like what I would need to adhere when I was 21, it would be the same thing. Because sometimes, again, it's kind of the similar, to your father giving you kind of that kind of the feedback. It's just like, it can be so hard. You just second guess yourself all the time. You're just like, is this the right thing to be doing right now? And just knowing that it will work itself out is sometimes all you need to hear to keep on trucking.
Yeah, I mean that's life, right?
Yeah.
We're way through the journey of life, and just go out and enjoy it, really.
Absolutely.
Yeah.
Well, Terry, thank you so much for your time. This has been a blast. I learned a lot, and I'm also very, very excited by the kind of the it feels like we're moving out of the Stone Age and we're in this sci-fi future now. And it makes me very excited for everything that researchers are going to be able to do with new technology like yours. So thanks again for the time, and thanks for coming on the podcast.
It was great to be here, John. I really enjoyed it. Thank you so much.
Yeah, thanks, Terry. Take care.
That's all for this episode of the Biotech Startups podcast. We hope you enjoyed our 3-part series with Terry Lowe. Be sure to tune into our next series, where we chat with Brian DeCaro, President, CEO, and Director at Sherlock Biosciences. Sherlock is enabling the democratization and decentralization of testing to personalize healthcare and make an impact on global health. Its proprietary platforms for DNA and RNA detection, powered by CRISPR and synthetic biology, will make rapid, accurate, and affordable diagnostic tests accessible to people at the point of need. Brian has a proven track record of funding and scaling businesses from venture-backed startups to profitable Fortune 50 public companies. His 20-plus years of experience, including his years of working in research and commercial fields, offer insights for founders to benefit from. The Biotech Startups podcast is produced by Exceeder. Don't want to miss an episode? Search for the Biotech Startups podcast wherever you get your podcasts and click subscribe. Exceeder provides research labs with equipment leases on founder-friendly terms to support paths to exceptional outcomes. To learn more, visit our website www.exceedr.com. On behalf of the team here at Exceeder, thanks for listening. The Biotech Startups podcast provides general insights into the life science sector through the experiences of its guests. The use of information on this podcast or materials linked from the podcast is at the user's own risk. The views expressed by the participants are their own and are not the views of Exceeder or sponsors. No reference to any product, service, or company in the podcast is an endorsement by Exceeder or its guests.